substances. So the idea is we can test in the tissue culture if our drugs are kind of be able to reverse Alzheimer phenotypes or whatever disease actually in the end. But this is only the first step, because this is cells growing in a petri dish. So what we want to do next, what is the next step, is take really a piece of tissue, for example from the human brain, a slice, grow that in culture, and have then a normal environment for the brain cells, because it's a piece of brain, and then test if those substances are working in those brain slices, in comparison to just cells are growing next to each other. And this in the end is the idea to then go...
Okay, hi. Welcome everybody to the lab. Maybe you should step a bit. Yeah, I think it would be easier for you to see. So this lab, I think, represents the highest part of the brain. So I hope Adi would not object for me.
No.
We are.
I like to be on the lower part.
Right, exactly. So he's at the lower part. And we are measuring, I think, probably the highest function of the brain, and that is how it is that we become consciously aware of the world outside us. Okay? Not only process the information, but also have awareness of what's happening outside. And I put in this demonstration for you to experience what I mean. So there are two pictures here which are flipping back and forth. There's a major difference between these pictures. And raise your hand if you see what's changing. Okay, one.
Yes.
Okay, there's another one. So it's not easy to see. Natalie, now point what it is. You see the pants here, changing from a jeans to like a fucking slacks. Now you cannot not see it, right? It's a huge, it's a major difference. But nevertheless, we were not aware of it. That's just an example of things that happen in everyday life when we cannot see big things that happen. We call this "Where are my keys?" which, you know, if you're looking for your keys on your desk or your glasses or your phone, and you look back and forth and look again and again, and it's just there, and you just don't see it until you see it, and you can't figure out how it is that you didn't see it in the first place, right? So we're trying to figure out what happens in the brain when you look at something. It goes into your eyes, goes into your brain. Nevertheless, you're not aware of it, versus when you are. So we do these experiments where we present this cluttered screen. And imagine that this is like a garbage pile, and you lost something in this garbage pile, and you're looking for this toy boat and this insect toy that is in there. Now, don't try to find it. It's a little bit small on the screen, so you won't be able to see it, but the subjects are able to see it if they look at it. And this is, we track the subject's eyes. We track the subject's eyes, and you'll see now how the eyes move on the screen. It's a little bit speeded, okay, but you see that this subject is very meticulously searching the image. Occasionally they will look down to see what it is that he's actually looking for. But he will go through these and not see them. Okay, and if at any time you would wait until it takes some time, and eventually he finds it and you can see the eyes darting. And at the same time, we measure the brain activity. Now, this is all looks like it's a little bit of a curiosity. But in fact, the reason that we are so interested in this is that there is a syndrome which afflicts patients who had stroke in the right hemisphere. A very frequent thing. And these patients behave as if half of the world just doesn't exist. Okay? Even though they can see, they feel the touch, they can hear, they do not respond or react to things happening on the left side of space when they have a right side damage. Okay? Now, this is a major problem for their rehabilitation. This is actually how I became a neuroscientist. I was an MD, and I did a residency in rehabilitation, and I met these patients and got completely baffled by these patients who are intelligent, speaking, understand exactly what we tell them to do, yet behave as if half of the world does not exist. They would leave the food on the left side of the plate when you present them with the food, because for them it does not exist. This is a lesion of one of these patients in an MRI, showing you this left part is where part of the brain was actually, died because of lack of oxygen. Now, when you ask these patients, for instance, to mark lines on a page, the patient understands the task, they mark all the lines, but they neglect to do something with these lines on the left. Or when we ask these patients, these are patients that I studied at the time, asked them to copy the star, the square, the cube, or the flower. And these are different patients' productions. You see, all of them forget some details on the left, as if they don't matter. So it's just to give you a feel, to rehabilitate a patient with unilateral neglect, it costs just as much as a liver transplant. Okay? So it sounds less heroic, but it is a major issue, and there are many more patients with unilateral neglect than patients who need a liver transplant. So it's a major clinical problem, and it's also a major question for basic neuroscience to understand how conscious awareness lives in the brain. Okay? And we use different methodologies to measure the brains of healthy individuals, humans, either patients or healthy subjects. This is an MRI machine which we have down the hill here, or a private MRI, where we can measure how blood flows to different parts of the brain when a subject is listening to something or looking at the screen and looking for something. It's a wonderful tool that we developed in the last 20 years. The major tool used in my lab is EEG, which allows us to measure the electrical activity emanating from the brain. And I put here this little mannequin with electrodes, the same one that we use, anywhere from 64 to 256 electrodes placed all around the brain, so that we can look at the brain from different angles and triangulate and figure out where in the brain things come from, and especially how fast they develop, because we can measure at the resolution of millisecond by millisecond the brain activity. Okay? And that allows us to figure out what happens in the brain. Here is an example. You don't need to understand exactly the details, but we're looking at the head from above. Okay, the colors show the strength of electrical activity. And these are healthy subjects. These are subjects with head stroke but do not have this syndrome that I described of unilateral neglect. And these are subjects with unilateral neglect, and without being an expert, you can see where is the problem. Okay? These patients here do not respond like the others when they're stimulated from the left side, and that allows us to tell something about why it is that they do not respond to the information. The most recent tool that we added to the lab, I already showed you an example of the eye tracking. So we are able to track the eyes with the same high resolution of every millisecond. And now we can do both. Measure the EEG, measure the eye movement, and figure out what happens in the brain when you look here, when you look there, when the subject looks at the keys or doesn't look at the keys. And that is a major advance. I don't know how much time we have.
2 minutes.
2 minutes. So let's step in. You can see our setup. Maybe we can even show you a subject moving their eyes, and you can see how we measure them. Okay? So please follow me. Okay, so just step in. So this is our setup. You can see our subject. Usually they would be with the come closer.
Thank
so
you, and I'll come ahead and
you can also look through the window. Okay. The subject. This is one of our lab members who volunteered to show. And we have this camera here looking at his eyes, and that allows us to synchronize his brain activity if he were using EEG at the moment. Okay, and get the information that we need. Okay? This is only a few labs in the world are able to kind of synchronize these two measurements together, and it's really kind of a revolution in neuroscience to be able to do that in humans. It has been done in monkeys and in animals, but doing it in humans allows us to ask these questions about the higher functions of the brain, which you cannot do in mice. You can't ask a mouse if he's aware or not aware of the information. Okay, unfortunately we don't have too much time, so thank you very much for...
Thank you very much.
Very strong walls on
it's a block sound.
sound.
It's actually a little bit scary.
Yeah,
to
this
be
is, you
inside.
can see him, this is where we want to get the subjects that are inside, and
I
this
used
is
to have
where
a
we check the eyes.
he would
No,
practice
usually
that
he has
with
the
no
hat
repeat.
with all the electrodes,
We
and
did, and I think
and
we were
also
lucky.
this only
that we
tracks
did.
the eyes,
Ah,
and the
okay.
hat
It's better
tracks
electrical engineer than
the brain activity.
Okay, thank you.
You're welcome. Thank you.
Welcome. Welcome.
They're still in, so...
Uno fue de aquí a Roma, primero 5. Y se perdió hasta Roma y de Roma
Pero ese vuelo de aquí para allá, ¿se fueron solo 5 personas en el avión? ¿O era,
No.
había mucha otra gente?
mucha más gente colombiana la juntaron en Roma, de Europa.
No, pero no
Sí.
pero no teníamos un vuelo para llegar a...
Okay, we need to let these guys pass
Yo sé
through,
que
so...
unos lo hicieron un viaje larguísimo. O sea, no fue directo pues, pero fue como... ¿Sabes qué? Ojalá que te pare, pues, más una ampolla.
Realmente extraño que vuelvas al laboratorio, la verdad.
Pero si quieres,
Porque recién mostraron
spécialiste de nouveau, ça vient des neurones qui nous doublent toute tâche.
Yes? I thought you were working on it already. Okay, welcome everyone. You can feel free to step into the lab because I wanted to see this over here. So, I'm repeating myself for the fourth time.
Last time.
Yes. Okay, so our lab is a bit strange because we're studying things that seem completely unrelated, and I'll try to explain how they can be related. So on the one hand, we're working on memory processes, and on the other one, we work on a population of cells in the brain that was almost completely neglected until now, and I'll explain. So my name is Inbal. I started my career as in psychology. So my bachelor degree is in psychology, and it took me about 2 weeks, I think, of psychology studies to understand that I must learn biology in order to understand anything about behavior that will really drive our science further and allow us to ask new questions is the integration of new tools. So we have a very big problem when we want to study the brain, and this problem is in many ways trivial. The brain is not transparent, and if we want to know what's going on inside, we have a problem because we cannot see through it. And we're using two different solutions here to overcome this problem, and I'll start with the easier one. But first I want to tell you about the population of cells that we study. At the very dawn of neuroscience, when neuroscience started, the person who kicked the whole field up, Ramon y Cajal, drew the first images of neurons and started a lot of excitement, and he also saw astrocytes and drew these cells as well, and then they said, nah, they're not important. And because he was the first, he was the father of the field, people took him very seriously and did not study astrocytes at all for 100 years. So we're already in 100 years delay. And when they did started to to study astrocytes, they did it in a very chauvinistic way. So people were willing to study how astrocytes are important. These are just glue, you know, they're not important to any function, which is completely untrue. We now know that astrocytes can do computations. We had a very big work 2 years ago showing that we can use astrocytes to improve memory. And this is in normal animals that have perfectly normal memory. You can still make it better by activating these cells. But the problem is that because for so many years no one studied astrocytes, we don't have the very basic building blocks that we need for our experiments. We know nothing about the distribution of astrocytes in the brain, their spatial interactions with different kinds of neurons. And for this, again, we run into the same problem of the brains not being transparent. So the first solution is very straightforward. We just take mouse brains and turn them transparent. Okay, so usually when you look on microscope slides in every movie where you see someone looking under a microscope, you have a glass, right, a piece of glass, and on top of it there's always a cover glass as well, and they seem to be completely attached to each other. In our samples, it's a bit different. We have the glass, and then we need to build this step, and the covered glass is here on the top. Because what's in here is a whole mouse brain. You can hold it if you want. So we have an entire brain that's rendered transparent, and now we can look straight into it and look on the cells. And of course, when you're looking at it with the naked eye, there seems to be nothing to look at, right? It's just a transparent brain. But some of the cells in the brain, we turned fluorescent. So we have cells that are fluorescent in different colors inside this brain. And now we can look on the entire brain or on entire structure. So in contrast to works from the past, that can look on 5 astrocytes, 10, there's one huge paper with, 20 astrocytes. We can look on tens of thousands of these cells, and you can see them here. So we're going inside the brain region called the hippocampus. It's the brain region that encodes memories, and you can see these star-shaped cells, and we can reconstruct each one of them, and now they're represented digitally in the computer, and we can start asking questions about the morphology of these cells, their distributions, things like that. And you can see that we have tens of thousands of them instead of very few. And one of the things that we can look at is the interactions between these cells and other cell populations. So for example, here, now the astrocytes are purple, and there's a population of cells here in green. They're called somatostatin cells. The name is not that important, but these cells, each one of them is hugged by an astrocyte. We don't know why. It's specific to this population. Other populations of cells that do relatively similar things are not hugged by astrocytes, so we don't know why there's this very special spatial interaction between these two cell populations, but we're definitely going to try to figure out why. So this is one way that we have around the problem of the brain not being transparent, just turning it transparent. Your hands are dead, right? It's a brain outside of an animal. And if we want to look on activity, we need to look at the brain that's inside an animal still, and we want this animal to be behaving at the same time. So we have, and I'm not going to show it to you, sorry, because there's actually a mouse in there, but we have mice that are trained to navigate in a maze, but system for mice. By the way, we actually also have a virtual reality system for humans, because the analysis of all this is done in virtual reality. It's much easier to look on 3D images this way. But we have also virtual reality for mice, and they learn to navigate in this virtual reality. And while they're doing that, we can look inside their brain. We remove, we don't turn it transparent, we simply remove the parts the activity of the astrocytes and tell you where the mouse is now in the maze. No neurons involved here. We're not looking on the neurons. And I'm sure some of you know that a few years ago Nobel Prize was awarded for the findings that there are neurons in this region, in the hippocampus, that can tell you where you are in space. These are called placers. So there are no place astrocytes. It doesn't work the same way. But and one last thing that I can show you that we can do with these clear brains is that while studying memory, we're not working only on astrocytes, but on neurons as well. And one of the things that's missing is the understanding of how different brain regions work together in order to create or recall memories. And having an entire clear brain allows you to see the connections between different regions. For example, in this movie, you see an entire mouse brain. Never mind which structure is where, but you can see all of these thin lines are projections, projections of one neuron in one brain region, going and talking, communicating with other regions of the brain. And this is, again, one of the powers of these techniques. Questions? You need
To breathe.
Yes.
Okay.
We need to study.
This concludes our full tour. I just want to mention that if in the future you think your delegates or the people that come from your countries are interested to visit us, we are always, one of the things that we put on our flag is to welcome guests, scientists, students, civilians, etc. So you're very welcome to contact us.
Thank you.
Okay.
My pleasure.
תודה רבה, ענבר. This is a shower.
Yes.